The MYSTIC trial is one of these trials trying to evaluate the opportunity of delivering immunotherapy frontline in patients with metastatic non-small cell lung cancer. The originality of the MYSTIC trial is that they propose two different immunotherapy versus chemotherapy frontline. So it's a 3-arm randomized trial. One of the immunotherapy is a dual inhibition of two checkpoints, the CTLA-4 and the PD-L1 with tremelimumab and durvalumab, and the other arm is only durvalumab versus the usual frontline platinum-based chemotherapy. This is the background for MYSTIC, but what is interesting is to keep in mind that, of course, the knowledge did accumulate in parallel to the MYSTIC conduct, and at the end of several discussions, there were 3 co-primary endpoints defined for MYSTIC: PFS and OS of the durvalumab and tremelimumab versus chemotherapy, and the OS of durvalumab versus chemotherapy. When I say it evolved over time, is at the end this endpoint only focused on the PD-L1 subset of patients with a high PD-L1, more than 25%, and patients were stratified for that. So the 3 co-primary endpoints, it's a challenge, but that was the primary design. We got to know at the ESMO Immuno-Oncology last year that the overall survival endpoints, the 3 co-primary endpoints, the overall survival endpoints in particular, did not meet statistical significance. It was interesting to see that there was a trend for the dual inhibition or the monotherapy in favor, in particular, a plateauing of immunotherapy, but it didn't reach statistical significance, probably also because having 3 co-primary endpoints does sacrifice some of the power of the trial. Numerically, there was probably a clinically meaningful change in favor of durvalumab versus chemotherapy, but it's just an exploration because it was not statistically significant. Based on that, we tried to look at better biomarkers. And in MYSTIC, the idea was to rethink the whole concept, looking at another biomarker, the one we discussed, the tumor mutational burden. Tumor mutational burden is a biomarker which now is being established as being predictive for immunotherapy in many cancer types. The problem is mainly to define the threshold in every disease, because every disease has a different median number of mutations, and also to have comparative data, because at the time being what we do is we take subgroups of patients and we compare renal cell carcinoma with high TMB and renal cell carcinoma with low TMB and see how they behave under immunotherapy. MYSTIC was an opportunity to look at TMB and compare treatment strategies in different strata of TMB. So really compare immuno and chemo, looking at TMB and stratifying for TMB. So we did it for tissue TMB, so we had kind of not a lot of tissue material, 10 slides per patient, but in these 10 slides we looked at the amount of mutations which were found there, and we did the same in the blood, in the circulating DNA. So to make a story a little shorter, we can now afterwards go into details. For the tissue, we were restricted by the number of samples. So only 41% of the patients in MYSTIC had this material available for the analysis. Based on that, anyway, and exploratory-wise, this tissue TMB was predictive of both the dual immunotherapy inhibition, durvalumab and tremelimumab, or the monotherapy durvalumab, and favored this option as compared to chemotherapy. That was a small subgroup of 41% with tissue TMB. We had, for the first time, a very large cohort of patients with blood TMB. It was 71 or 3, I don't remember, % of the MYSTIC trial patients who had this biomarker available, meaning more than 800 patients. And this could be more formally evaluated as a predictive biomarker for durvalumab and tremelimumab. And we looked at different thresholds, because you can have many thresholds of TMB. The idea was to define a threshold which was showing a benefit of dual inhibition immunotherapy, and maybe even a monotherapy durvalumab. And very importantly, a threshold which would explain if there is a difference between monotherapy and dual inhibition, double immunotherapy. And at a threshold of more than, we call it 20 mutations per megabase in the blood, all these objectives have been met. This threshold was allowing to predict the overall survival benefit of durvalumab with a hazard ratio of something like 0.7, and to a wider and broader magnitude and effect, the overall survival benefit of the durvalumab and tremelimumab arm with a hazard ratio almost never met, or 0.49. So suddenly you can differentiate both, but both are statistically better than chemo for survival, right? But more importantly, when we looked at this very favorable durvalumab, tremelimumab in high TMB, we could also show a PFS benefit with a hazard ratio of 0.56 and a response rate which was almost doubled. So really meaning that by selecting enough, because it represents this high TMB, maybe 20-25% of the patient population, by really selecting the top percentiles in terms of TMB, you can really show a complete picture of benefit in all the outcomes which define a clinical benefit in a clinical trial. So the idea was probably you have to aim at a high threshold, and then you can really decipher and describe a benefit of immunotherapy in these patients over chemo. To evaluate TMB, tumor mutational burden in tissue, you need quite a lot of material, and a good quality material, because it needs a certain number of cells, a component of tumor cell which is viable and to be analyzed. So whatever the reality of the future, there will always be patients where such a material is not accessible. It's mainly related to the difficulties and invasive nature of getting a biopsy, a second and a third biopsy. Beyond the difficulty to get the biopsy, it's also lots of time. If you do a biopsy, you have to reorganize a second one. So in terms of turnaround time, this becomes to be a difficult biomarker, invading very long. That's why we like the blood. We have to be also very realistic. There will not be enough DNA in the blood of all patients. In MYSTIC, what we observed is probably 17 to 20% of the patients had not enough DNA shedding in the blood to measure anything. So we'll never get BTMB in all patients, but the vast majority. And what is good is that the turnaround time and the invasiveness of such methodologies are all very relevant, because it's short and it can be accessible just by a blood sampling, right? So this looks like being a biomarker which is very interesting in using frontline and very efficient to do so. Many companies are competing in developing it. Many, but I think the most important is to make access, to get access to these strategies, just to be able to offer it to all patients, right?